Hello, everybody. Welcome back. We're going to talk about the glycolytic pathway now in this video. The term glycolysis means splitting of glucose. So glyco means glucose, lysis means splitting. And so in this process, we're actually going to split one glucose molecule into two 3-carbon molecules called pyruvate. And you can see this over here where you're seeing glucose here, and way down here we have 2-pyruvate. So glucose is, the chemical formula is C6H12O6. And the chemical formula of pyruvate is that's not a C. C3H5O3, and we have 2 of them. So you can see that we keep the carbons the same. 2 times 3 is 6 carbons, and we keep the oxygens the same, but we remove some hydrogen in this process, okay? So we convert glucose into pyruvate. In the process, we're going to need some coenzyme carriers to pick up hydrogen because we lost hydrogen from here to there. And so glucose is oxidized and NAD is reduced. You can see it right here. Here we have glyceraldehyde 3-phosphate, and here it's showing the production of NADH. So right here, in this step, we're removing hydrogen ions and electrons. That's oxidation. We also produce ATP using substrate level phosphorylation. So remember, substrate level phosphorylation is the process of adding a phosphate directly to ADP to produce ATP. And this is how we produce ATP in the glycolytic pathway. So we start out with an energy investment phase where we have to take and add in energy. So we use ATP to activate the glucose, which then is going to split. So glucose is activated, it becomes glucose 6-phosphate. It then is converted to fructose 6-phosphate, then to fructose 1,6-diphosphate. Eventually it is split into glyceraldehyde 3-phosphate. And then it's going to lose hydrogens and electrons and phosphates, and it's going to eventually become pyruvate. So the energy investment step is we add in 2 ATP. We produce a total of 4 ATP, but we only get a net gain of 2 ATP because we use 2 ATP in the beginning. The transition step then is a middle step, or a step that moves the pyruvate to the citric acid cycle. So pyruvate is a 3-carbon molecule, but we need to remove a carbon from the pyruvate to allow it to bind to a 4-carbon molecule in the citric acid cycle. And so the transition step, just its main job is to remove that carbon from pyruvate. In this process, we're going to also oxidize that molecule further. So we are going to need NAD to pick up those hydrogens and electrons. This is oxidation, or this is, I'm sorry. Let me do this right. So pyruvate is being oxidized and NAD plus here is being reduced to NADH. When we take carbon out of something, we decarboxylate it. And so here we need to have oxygen available. So oxygen is also needed. We do not use oxygen in glycolysis, but we do use oxygen in the transition step as well as in the Krebs cycle. Once we have removed the carbon and produced carbon dioxide as a waste product, once we have removed the hydrogens and electrons and produced NADH as a taxi that will move on to the electron transport chain, we then mix the acetyl group that's left over with coenzyme A, producing acetyl coenzyme A. And that's going to move into the next step of aerobic respiration. So at this point, the glycolytic pathway occurs in the inside of the cytoplasm of the cell. Transition step in prokaryotic organisms occurs in the plasma membrane of the cell. In eukaryotes, this occurs in the mitochondria. I'm looking at time. And I'm going to quickly, actually, I'll stop here and we'll get into the Krebs cycle and electron transport chain in the next video. So I will talk to you guys in a little bit. Bye.